Welcome everyone to the face-to-face meeting and all of those who are online. And it is my great pleasure today to introduce Professor Ronan Lyons. When I was appointed as a young professor at Swansea University, my head was full of how important it was to find biomarkers and genes and do the preclinical pipeline, make sure that we could go into the future with impactful trials. But it was my work with Ronan and his team in the Health Informatics Research Unit, which then became SAIL and then became a HDR UK Centre. And it showed me the value of the new epidemiology, where you were looking at data points at millions of points of contact, not 100 or thousands of points of contact. And it really does change the way you look at data, the way you analyze it, the way you interpret it, and the way you implement it. And so it is with great pleasure today that I am going to introduce Ronan. So Ronan is Professor of Public Health at Swansea. He is a graduate of Trinity College, Dublin, and he has developed the cohorts and population cohorts in the Secured Anonymised Information Linkage, or SAIL. And you'll hear a lot about SAIL and the studies that they undertake. So he has held many senior roles, including with the MRC, which is the NHMRC equivalent in the UK, and senior leadership in the COVID-19 data linkage projects and supporting the UK in its response to COVID pandemic. He's Associate Director of the MRC Dementias research platform. And it really does show the strength of infrastructure in health sciences in that you can transcend the medicine map when you can take on responsibilities in neurology, in cardiology, in fragility, in multiple morbidity, etc. And so Ronan has been very influential in the neglected field of injury prevention and has chaired the US National Centers for Health Statistics and an international collaboration effort in injury statistics and methods. He was awarded an OBE in 2021 for services to research, innovation, and public health, and he is a fellow of the Academy of Medical Sciences in London. And so it is with great pleasure that I now introduce Ronan, who is a great friend and colleague, and also a mentor of mine from Swansea, and it is a great pleasure for me today to introduce him. Thank you, Ronan.
So, So thank you very much, Mark, for that introduction. You're not finished yet?
I haven't.
No.
So, the next thing I'm going to do, of course, is acknowledge country. So we acknowledge the traditional custodianship and the law of the country on which we meet today. And today we are at the Gadigal peoples of the Eora Nation. And I give my respects to elders past, present, and emerging, and remind everybody that healthcare has been taking part on the land and on this land for nearly 60,000 years. And thank you very much for that. And then a bit of housekeeping. Presenter questions, please, at the end of the talk. And please put that into the Zoom function for our online participants. And if you need assistance, please let us know during the course of the talk. Thank you.
Right. Super. Well, thank you very much for that very warm welcome. It's a great pleasure to be here in Sydney. It's great to see lots of people coming to hear what we've been doing in Wales and the UK, and also a lot on our international collaborations. So I'm going to talk about essentially what's on the slide here, about how you actually conduct research and how people can access data right across the world. And I'll be talking a fair bit about the SaIL data bank, as Mark says. So I've got lots of slides, so if we run out of time, we'll just stop, but we'll see how we go. All right. So one of the things that comes up in this concept of trusted research environments, and I don't know whether that's a term that's used widely in Australia. It's used very widely in the UK. And so we'll talk a bit about what TREs are, how the ones that we've created work, and then give some examples of other ones and the potential for collaboration. So this was just a sort of introductory slide created by other people. TRE stands for Trusted Research Environment, and these are sort of data safe havens. Now, they're complex things that operate through a thing called the 5 safes, which are mentioned here on the left-hand side. And it's about how you sort of develop systems that allow essentially people who've been accredited and projects that have been externally assessed access to sort of widespread data and allow sort of deduction of outputs without essentially risking anybody's privacy. So they're really important because they've enabled us to do all the stuff we're doing. They are sort of growing. In the UK, essentially, there are 5 national ones. So the UK, United Kingdom, is to some extent disunited because the health systems in England, Scotland, Wales, and Northern Ireland are completely separate. And whilst they collaborate, essentially, there is a bit like the state system here is they control their own data. The Office of National Statistics has a different one. So I'll talk about, particularly on SeRP, on how that works for us, really. So the presentation, the next part of this is really about this secure eResearch platform that colleagues have created about how we basically bring data together, share it with lots of people, with the people who are the data custodians remain in control. So the first thing I will say about this is this is essentially team science. You need lots of different disciplines, people from different agencies, lots of public engagement to make it happen. For lots of people on this slide, it's probably only about 1/3 of the people who sort of work with us. I would just point to the top guy wearing the suit up there, Simon Thompson, our Chief Technical Officer. Simon is the brains behind all of the technology and operates this. He's massively important. Many other people are important there. On the bottom left-hand side is Jan and Chris Davis, and they are members of the public who help steer everything we do. And I'll say quite a bit about public engagement and how that helps us as we go along. So essentially, well, what is the sort of SeRP thing? It basically has been bolted together over time by investments that we've had in various UK research centres of excellence. So we've had some on CLIME, which is about microbiology data. The Medical Research Council, there were different iterations. The most recent is the Health Data Research UK. And then from the Economic and Social Research Council on administrative data. So essentially they are publicly funded research cloud, designed to use and protect the use of secondary data. We do operate them on a cost recovery model because it's not all funded. And essentially they have evolved and continue to evolve depending on what the researchers need. So this is one of Simon's slides. The number 28 there is important. So we basically, SeRP facilitates 28 separate tenancies at the moment, all right? So lots of them are listed here. I don't think all 28 are listed. Then there's the usual sort of funder logoria over on the left-hand side. We acknowledge all of the funding of where it comes from. I will say a fair bit about SaIL, which is about the routine health data, and I'll say stuff about some of the other ones that come along. They actually hold different types of data. Some of them are routine data. Some of them are cohorts. Some of them are different types of data. So in not just coded data, but text data and narrative data, and all sorts of different things. And at the bottom, we also have a separate GIS system, which a guy called Richard Fry runs. And that allows us actually to link data to individuals at the household geospatial level without us ever knowing who the people are or ever knowing where the address is in the design. So they all have different governance approaches because essentially each tenancy controls what it wants to do with data and how it allows people to access data. So the Sale Data Bank, which we'll talk about a bit, we have this independent governance review panel. And that's made up of people who are all outside of Swansea University. They are members of the public. They are data protection officers from the health service and people with expertise in those areas and ethics. And they get to see every proposal that essentially is up for using the data, and only things that they say can happen do happen. I will say a little bit more about that. The Dementia, UK Dementia platform is a platform funded by the Medical Research Council, which brings together data on many different cohorts with essentially a view to changing what we know about dementia and what treatments are developed and how effective they are. The Administrative Data Research Network is really about non-health data, but we do actually link it to health data. And the MS Register is a platform in which we've actually actively recruited large numbers of people with MS from around the UK, largely through clinics and neurologists, to provide multimodal data and engage in research. And the CRIS network is one we operate for a South London mental health trust, and they deal very largely with identifiable narrative clinical text. So we have, apart from the UK, we have expanded a bit out into Canada and Australia. So we run systems in Vancouver for the health service. And so far, we also have deployed the SeRP technology in Monash University through this Helix thing, in University of Western Australia. And yesterday, I was at the University of New South Wales. They've signed up to a SeRP development there. And we would hope that to be able to expand this across Australia through, there's another grouping, which I'll come back to now in a moment. These drawings, which I'm not going to go into any detail, just sort of start to show some of the complexity about how essentially a researcher starts from one end, gets access to data and outputs at the other end. It's very complex, as you can see, this is one of Simon's slides. 14 firewalls, 2 million entries a day, but it works really well. And the researchers don't see virtually any of that sits behind it. One of the things that's also changed over time is the regulatory compliance with holding data. So there are lots of different organizations that we have to receive approval from before we can do what we do. And we have many different assessments. The main ones are up on the screen here. We've been approved by all of those. We have penetration tests. We have all sorts of things. So actually running the governance of all of this is quite a complex business. And but it all comes down to essentially this sort of 5 safes approach, really. So everything that's done in the system effectively is a project, and sort of there are roles associated with projects, and then there are permissions associated with those roles. And then people have contracts about what they can and can't do. And then there are resources which are including compute and data and some assistance that go to all the projects. So Simon, again, has created this system that controls it all, I think called National Research Data Appliance. And that basically runs the system. An awful lot of it is automated at this point in time. And again, he comes up, I love his title, his slide, a simplistic diagram on the left-hand side. Well, I don't think anybody would say it's simplistic, but it basically is how the user at one end eventually ends up with the data and all the technology that are used at the other end. And one of the things, when we started off, we used basically largely IBM DB2, but we now have instances of almost every technology that we deploy, and it's constantly changing. And we do that because researchers want different technologies, and we find a way of making sure that we can service all of those. The vast majority of what we do it's done remotely on servers at Swansea University, but on the bottom left-hand side, you can also see that we have a burst-out facility into the commercial cloud from Amazon Web and Azure. It's not used massively. People find a lot of the in-house compute works as well, if not better. And also, we have to charge the commercial rates that they charge us. And they're about 3 to 4 times more expensive than what we do in-house. So mostly people say, well, do you know what? I can do without that. Now, I understand that in-house versus commercial cloud facilities in Australia might have different price levels, so that might be a bit different, but that's our experience. So essentially what we run, if you like, is a remote access facility in which all the analysis is done under our control. It's accessible through the internet. There is a safe room thing that fits some of the data. And we have large numbers of different analytic tools depending on what people want that are mentioned there. We have run various workshops and hackathons to actually help people use the data and bring new people involved. We've done most of those through the Dementias platform and the Alan Turing Institute, which brings in lots of different mathematicians. The thing about a lot of this, though, is that you actually need people from multiple disciplines involved in any project, because actually understanding the data and the complexity of the data is quite a major challenge. And we have metadata for most of it, but even still, the nuances in data sets need to be understood. It's quite easy for 2 and 2 to add up to 22 instead of 4. So a bit about the Dementias platform, which I'm associate director for. That essentially has brought together many different UK cohorts. We've been working on common ontologies for harmonizing some of the variables between cohorts. That is a really difficult task. I see a gentleman at the back taking a picture. You're very welcome, because I'll leave a set of the slides here and you can have them. So you don't need to take pictures if you don't need. And cataloging. Increasingly, we're working on people's interests are in multi-modal data, particularly linking the cohorts to genetic data, imaging data, and down the line, more and more routine follow-ups. So again, we have different types of infrastructure and cluster computers and different facilities for managing imaging data and stem cell data, which are coming into this system. So the imaging network, there was another investment by the research councils many years ago, and they basically put 1 and 3 Tesla MRI machines in many of the major universities. They all basically are powered by XNet, and they have separate instances of those. The main centers, geographical centers, are there on the screen. We basically now are pulling all the data from those into DPUK, into a single system to enable researchers access the data and carry out analysis. And again, the technology of that changes. So on this slide as well, we were also involved in the Australian Research Data Commons, and I sit on the steering group of that. And that has sort of helped with some of the spread into Australia. So part of that is that this is going to link also into the Australian Imaging Service, and that will make a difference. One thing I would say to you compared to other things, many other things, the Dementia platform in the UK is well worth exploring if you're interested at all in dementia. And one of the, apart from the brilliant data that are available, the other thing, it's completely free. It is funded by the UK Medical Research Council. And for people who got approved, not sure quite what happens on the screen. Oops, there we are, let's jump back. People who got approved projects access the data for free. Other things do have charges, so that's well worth looking at. And also, there's quite a lot of development about how we handle genetics at different levels and the compute that's supporting that. So lots of things going on in those areas. Again, you can see the sort of technology and that Simon is bringing on board to facilitate the analysis of such data. So say we're very flexible in that way. We look to see what researchers want, and we implement that to keep them all happy. So lots of things happening there about how all of this changes in that. So I'm going to sort of switch a little bit now and talk about some of the projects that are there. As I mentioned, there's a thing called the UK MS Register, and that's run by a colleague of ours, who's recently promoted to Associate Professor Rod Middleton, who started out life as an ITU nurse and now runs a whole MRC, or register funded by the MS Society. So these are some of his slides. And what they've been doing is they've been recruiting many thousands of people with MS from right across the UK. And then they bring together the data from those people through they have permissions to link to all their clinical records. They are getting tissue samples from people, which are coming into a tissue bank. So there's genetic data, there's also imaging and MRI data, and there are data collected by people on the responses when they send out questionnaires. So I think it's probably the best systems that I've seen working so far. And again, they use this to do all sorts of different research now. And this is from a fairly recent publication looking at what happened to people with MS during the COVID epidemic. And basically then looking at what treatments people were on and whether those disease-modifying therapies made a difference or not to whether people ended up in intensive care or dying. So essentially, the lines are there for each different drug set that were used. They basically got dashed lines, which are people who've had confirmed COVID, but they've also have solid lines for people who had suspected COVID. You may remember when the whole COVID thing happened, we didn't have enough testing available. So in the early phases, there was quite a lot more of suspected COVID than there was of confirmed COVID. And it does show some differences. I think the differences aren't quite so important at this age, but it's just to demonstrate what they can do with the data and the treatment. Another area of work that they've been looking at is some behavioral issues and what happens. And this sort of study that they were looking at here is looking at people who smoked and who stopped smoking or continued smoking and how that affected the progression of MS using some of the MS disability systems, which are on the left-hand side. And basically what they've shown that actually clearly the people who stopped smoking, their MS progressed less slowly than people who continued smoking. So it's quite an important finding, that one. They've also been having a look at about how can you actually monitor the incidence and prevalence of MS. And they've been doing this in Wales, looking at what they've got that's reported from people and what's available in the routine data. So the routine data would include both general practice data and the hospital discharge data. So on the left-hand side, it's been looking at over time is how has the incidence changed over time? And over time, they've actually shown that there is a gradual reduction in the incidence of MS, which is interesting. The middle slide basically shows the sort of the age distribution of the prevalence of it, and then how it's changed. And again, how this is changing over time. So whilst the incidence has gone down, actually the prevalence of the condition has gone up. A lot of that is due to the fact that people are living longer. So we've got greater survivorship, which is a good thing. Another thing that they were doing, that there was a big sort of study done on the general population about the Great British Intelligence Test. Now, people might say that's an oxymoron, but anyway, it was done on hundreds of thousands of people through the BBC, and data were made available. And the MS group actually went and recruited their people to do it also. And so you can look at the distribution at the bottom left graph between the UK population and MS patients. And it does show that there is quite a cognitive difference between the two groups, which is quite concerning in that area. They also spend a fair bit of time in trying to work and find people with epilepsy, and Mark is involved in some of these studies. And again, basically looking at how well does that work when you combine diagnosis with anti-seizure medication. And you can sort of see the sort of sensitivity and specificity that we get when we're using that approach. And that was about nearly 9,000 people that were followed up over a 6-year period. So prevalence of 0.7%. And there we have some incidents as well. Another part that people spend a fair bit of time looking at is the relationship with deprivation. So this is a map of Wales, relatively small country, 3 million people or so, and we have small areas, and we basically each area is about 2,000 small areas, gets put into sort of fifths of deprivation, and we can look at whether the incidence is higher or lower than that. And we do, of course, have higher incidence and prevalence in deprived areas. It is associated. There is always a business about, well, do you change your location when you're ill? The sort of social drift and social causation one. And again, so here we have, like, on the one hand, on one side, we've got the prevalence of epilepsy by tenths of deprivation and the incidence by tenths of deprivations. So there are clear gradients there. But essentially, not vast changes in where people have been moved to. Because one of the things we could do in all of this over time is track everybody's movements. And another area that they've been looking at essentially is the relationship between cardiovascular events and epilepsy. And there is a sort of a clear relationship, as you can see on the left-hand side here. Just running this, yeah. So these are about unadjusted and adjusted. One of the other areas that since they've been looking at that, then saying, well, does essentially this relationship, is it affected by therapies? And so whether we've got enzyme-inducing or non-enzyme-inducing treatments, what difference does that make? And actually, you can see here, really, it has very little effect, which is good news. Another area they're looking at then is about the use of these drugs in pregnancy, which of course is controversial, and you have to balance essentially what's done. So there are some known risks with anti-epileptic drugs, and there are also people who are concerned about long-term effects. So part of trying to understand this is that a lot of our coded data is not brilliant. And essentially very few things that actually that are in clinical records actually get coded into hospital discharge. And this is where essentially the group spent a fair bit of time working on natural language processing. So you basically, these are working with clinicians, so we get access to the letters, and there's essentially training sets, and then that basically goes into a set of technologies that are used to assess it. And then there's some validation work, and the validation is done again by clinicians. So we can basically understand how well, if you like, the machine learning, natural language processing works against the clinical expertise. And it works well. So here it is, if you like, just sort of running through what the sort of letters tend to include. There's a guy, Owen Pickrell, who leads this, who's a neurologist who specializes in epilepsy in Swansea. And he's also got a network of all the epilepsy specialists in Wales. And we've recently just got a grant to extend this work substantially, which is very nice. So you can see essentially on the letters, you can see there's terms there that help tell you whether or not the epilepsy is confirmed, about what sort of seizure types there. You know, EEG results, things about frequency of seizures, medications that have been used in the past, side effects, and the current medications. As I say, the vast majority of these things do not get coded into the coded hospital discharges and do not get coded into the general practice records. So there's a lot in those, but you also need to go for things about this. The other thing is that they have been successful in getting genetic samples from lots of patients with epilepsy and sort of linking the genetic data in SaIL. And so this is one of the first examples of doing that. The sample sizes are still small, so none of what you're seeing here on the screens are statistically significant, but it shows the capacity to be able to bring in and analyze genetic data on the system. So I'll turn, I've mentioned SaIL a bit there now, and I'll say a little bit more about that. So I wrote a grant in 2006 for £5,000 to actually put together a group to think about creating one of these trusted research environments. And that was successful, and we got further funding. And so the SaIL system was effectively set up in 2007 and 2008. We spent 2 years planning to do it. Part of that planning was to identify anything that might go wrong, and to design that out of the system. And so that's worked very well. We have not had a data breach or any problem, any major problem, since 2006, 2007, when this was designed. We have huge amounts of data that come into the system because it's trusted, and it's very automated, and it works very well. Now, we don't just have Welsh data in that, because as I said, there are many other data sets that we also host there. So it followed this 5 safes concept about safe projects, safe people, data settings, and outputs. The system works by the fact that we don't hold any information on a person's name, address, date of birth, or geolocation. Files get split into 2. One is sort of identifiers, and they go off to a collaborator of ours, which is in the health service, and they do the identity matching and first stage anonymization. And then the content of the data flows to us. So we've got lots of different things that to make this happen. That's just effectively a bit of diagram you can look at your leisure about how the split file system operates. They encrypt the data before it comes to us. We also encrypt the data as it arrives. Nobody holds both encryption keys, and then many of the projects are encrypted a third time. So you do have person-level data, but no clear identifiers attached to them. I said one of the things that we learned early on is to develop a lot more public engagement. And so we have a consumer panel, which are made up from members of the public. And that started in 2011. And they're really important people. So every proposal they get to see. And they look at all the proposals and they have two questions in their mind. One, is this in the public interest? And secondly, have the people taken sufficient action to protect privacy? And they also provide feedback to the researchers on how to write things properly in standard English that normal people can understand. And they're fantastic. And many of them then have been involved in many of the projects and helped co-design them and explain the results to their communities. So here we are at the moment. We've sort of more than 850 users. The vast majority, as you can see, are in the UK, but we have people in the US, in different parts of it, I think in Hong Kong, and in some of parts of Australia. And one of the things I'm hoping is that we can have dots for Sydney not so long down the road. So it's worthwhile having a look at what you do. There are some charges on using the data, but it is not expensive compared to many other things. We do run a whole pile of analysis about what sort of areas of research is supported. And you can see there on the left-hand side one, lots of different areas. This facility allows things support. We have an upward trajectory of projects. The drop-off you see at the end is just because that's an incomplete year. And we can also look at who has access to data on the left-hand side. So 3/4 of it is academia, but we do also have health service people, we have government social researchers, and we have some businesses that have access to the data. Those are very well controlled. And I'll talk a little bit now about the data linkage during COVID. So I wrote a grant to the MRC and we got funded to link the entire data on the Welsh population of the COVID epidemic. And our team was seconded to work with our chief medical officer at that stage to undertake rapid analysis of data to inform policy. So that's the sort of protocol there of the paper. On the left-hand side, you can, on the right-hand side is all of the data that came along. This is the first time we were also given permission to link in the national census from 2011, which is very useful. And we have permission to bring in the new 2021 census. So many, many different data sets. I'd point to this one here, is that during this time also, we got permission to bring data in from what's called the Wales Results Reporting Service. And that is every laboratory test on every person for everything. It's absolutely massive. It's billions and billions of rows, but it allows us now to get to a depth of analysis that we previously hadn't been able to do so. And we do have embedded trials and cohorts in all of this. So there were lots of different publications. I mean, I just put some of them up here. Early on, we published quite a lot on medRxiv before they went to main journals to release what was happening. Some examples there. You can see some there on what's happened in care homes. I'm not sure whether I'll go into that in any great detail. We had pretty much disaster in our care homes. COVID effectively doubled the mortality that we used to having. So a lot of people died in care homes. The bottom left-hand one was particularly interesting in that in this system, we were able to also bring in data on all school pupils and all educational staff. and we linked all the children to all their household members and to their schools and their teachers and their household members. And that allowed us to look at transmission patterns across schools and that. Now we had, you know, the odds ratios for a household contact, a person being positive, creating another one is very high. It's about 40 per person. But you're also able to see in this system that when a teacher had a household contact who was positive, but the teacher was not positive, we could still see an increase in the kids that the teacher thought. And the reason for that is a lot of this, of course, were asymptomatic. So there were lots of asymptomatic transmission going on. I'll talk a little bit now, coming towards the end, about some of the mortality. We were asked to do some work for the Royal Statistical Society. Each of the 4 countries was about what was happening in their country around mortality. And this work is really largely done by Rhiannon Owen, who's a fantastic statistician in our group. So what she's been doing here essentially is, this is our standard mortality from 2016 onwards and the epidemic here. And essentially you've got black lines, which is the actual, you've got expected lines, and then you've got red dots for things which stood out, right? So the first thing people asked the last time I did this presentation, why does it go up and down so much? And the reason it goes up and down so much is that we have terrible excess winter mortality. We've got huge seasonal patterns in death in the UK. That's due to absolutely crappy housing and cold housing. So we compete with New Zealand to do this, do housing badly. If you go to Scandinavia, where the houses are more properly built, you don't see the patterns. They're not as much as that. So there are lots of seasonal patterns that put in. So the 2018, we had some excess mortality. And that was the last flu epidemic that we had. And then you can see the dotted line after COVID, where we've had quite considerable excess mortality. And then on the right-hand side, you're looking at essentially the mortality patterns. So the solid lines are all cause, and the dotted lines on the bottom are essentially COVID classified mortality, which is substantially lower. And again, you can see how that changed over the different periods at which the different variants were there. So one of the things that also changed in this was where people died. So these again are essentially deaths within hospital and deaths outside. UK, about 2/3 or 70% of the population die in hospital. But during COVID, then we had sort of massive increases of people dying at home. Not all of them were in care homes. And again, we've been able to look at how this differs essentially in, if you like, the white population on one side and the Asian population on the other side. This is where the census linkage was really helpful, because our coding of ethnicity in health service data is not great. And there are differences in the mortality by the different variants, by the different ethnic groups. If we look at not just the sort of COVID-related death, but particular different causes, these ones are on the right-hand side respiratory and the other cardiovascular. You can see we've had excess mortality across both of those since COVID. And if I keep flicking through and say neoplasms, so cancers on the right, and digestive disorders on the left, excess there as well. If we do some by mental health, but on the other one, the trauma one is interesting. So we had excess deaths early on during the pandemic from trauma. We haven't quite worked out what's happening there. A lot of people were at home. People were not moving around as much. People may have become more deconditioned and had more falls. It is something that needs further explanation. As well as doing all of this in Wales, we increasingly got involved in UK-wide collaborations across the four nations. So this is a paper that isn't out that long. It's just last month, I think, about how well the vaccines are working and waning. And this was putting data together on 12.9 million people across England, Wales, Scotland, and Northern Ireland. So it allows us to do things like this, which looks at, apologies for the slides, but these are the slides you download from a published paper. They're awful, aren't they? But you have to use them. So you can see essentially the line of no effect with one, the black line going across it, and anything below that is protective. And essentially, this example and this one, this is a Pfizer one, is that that protection lasted till about 70, 75 days. And after that, we have waning. So it allows us to actually see how the different vaccines are doing, how the boosters are doing, and there's a lot of work that's continuing with that. So that's where I'm coming to an end now, to leave a little bit of time for questions and that. I mean, these technologies, I think, do provide huge opportunities for research. We are very keen on international collaborations, and particularly about how you can learn and share between countries. Some of us are also very keen on the evaluation of natural experiments, in which different policies and interventions in different places have different effects. So we like to collaborate in those. And also the opportunity to have meta-analysis of larger scale observational data. So the creation of the SeRPs in Australia, I think, is going to help a lot, because actually doing things on similar technology stacks is a lot easier. And as I say, I'm very happy to put people in touch with different research teams to see how it goes. So I'll stop there. Thank you.
Thank you, Ronan. That's a brilliant summary of the wonderful data infrastructure that we enjoyed when we were in the UK. And I think it's just the way in which clinical trials and biobank and imaging all plug and play these platforms. It really is paradigm. So I'm just going to open it up to questions. We'll start from there, and we'll also see if there's anybody online as well. Sure.
Thank you, Professor Lyons, for a very interesting presentation covering many different topics. One topic I found interesting in particular is epilepsy and how the incidence rates differed by socioeconomic decile, which made me wonder two things. First, I'd always thought epilepsy was primarily a genetic condition, but it seems from your findings that there may be environmental factors as well. And I wondered, had there been any research into how environment affects the incidence of epilepsy? And second, just wondering, was that disadvantage level, was that computed from the postcode? You mentioned that you try to protect patients' confidentiality by not having fields that are readily identifiable, for example, date of birth and postcode. Just wondered, are they in the data and just not given to researchers? Or?
Right, yeah. So if I start at the end and go backwards, this essentially full addresses go off to our NHS collaborator, who has a list of everybody in the country, because we got a free to use NHS, and you go in at birth and there. And so they basically then will match the individual against their NHS register. And what they'll do then is they'll essentially create a unique anonymized number for that individual, but also they'll have a unique anonymized field for the geolocation for that person. And so the geolocation actually happens at their address, and then that's brought together into small areas called lower super output areas. There's 2,000 of them. Those are divided into fifths or tenths, which we standardly use. And actually, but what the researchers will see is the fifth or the tenth. So all that other stuff happens automatically in the background and enables researchers to do sort of analysis by social position and deprivation without actually seeing the raw data that underlies it. The etiology of epilepsy and that, it's not my field of expertise. I know a little bit about it. Mark, it's far better to be able to answer that.
So, yes, epilepsy is genetic, but it is a combination of monogenetic and polygenic inheritance. But remember that what causes seizures are triggers like sleep deprivation, alcohol abuse, drug abuse. And so what we're seeing is, and we always thought that the diagnosis of epilepsy meant that you lost your job, you lost your ability to drive, you had social drift back into poorer areas. And that was driving the incidence of prevalence. But it's not. It's social cause, it's causation. And it is the lives that they live in deprived areas with high sugar, high fats, high oils, high salt diets, with drug and alcohol abuse increased. And it is all the triggers, the environmental triggers, that then get those seizure patients into hospitals and into clinics. So it is causation. And in fact, that is the one study that went straight into the Welsh Assembly government, straight into policy and straight into implementation. And I think, that's one of the things. But I think we've got to get away from thinking that epilepsy is just genetic, because all of the consortiums and all of the genomes that we got back from the Broad Institute, around 30,000 people, is showing that there is a genetic contribution, but it's not the full story. It has to have environmental components.
And I mean, deprivation affects many different things. So you'll have intergenerational deprivation. The babies born in the deprived areas tend to be more premature. They tend to be smaller. So you've all of that comes through into the development of many different conditions.
Thanks. I'm going to turn it on first. I'm going to ask a question from online. So Mark McLean asked it. It might have been a little bit premature when he asked it. So I'll add an extra question on the end there. Said, fantastic presentation. Thank you. Can you comment on the availability of SeRP for Australian institutions? And he said he's working on the HASANDA project with ARDC, Australian Research Data Commons, and not being used by many of the participating unis. And then I'm going to add a second question on for myself, which is about the dynamic nature of the databases and how often they're updated in the system.
Yeah, so early on, we had an instance of SeRP in Monash University. And the reason that happened is because I've been collaborating with Belinda Gabay on trauma outcomes for decades. And she was a frequent sort of visitor to Swansea and would spend some time every year. I thought, well, I really like this data system and how it's working, because she did quite a lot in it. And then she sort of persuaded Monash to say, we need to do some of this. And they were doing some themselves, but it didn't work tremendously well. And then they decided that they would sort of co-invest in its development. So there has been a SeRP instance working in Monash for a good number of years. And then other people were having a look at, well, how well that's working, and it's working well for their research community. So the Australian Research Data Commons thought, well, this is something we'd like to see actually expand more across the Australia. And so, there is now an instance working in University of Western Australia. And I heard recently, well, yesterday, University of New South Wales have invested to do some, and we're hoping that others will join in that consortium. Because the other thing about this is we also know, data sovereignty is a really important thing, and you need to be able to stay in control of your data. But at the same time, if you can develop a system which facilitates collaboration and meta-analysis and federated analysis of data across system for greater scale, then you get involved in much bigger research projects that have greater impact. And so that's what we want to see happen. So there is a group working on how can we do federated analysis? And some of the discussions at UNSW yesterday was about imaging. Could we have some common tooling and analysis of imaging in the UK, imaging in this part of Sydney, and then actually allow the research community access to it, even on this sort of distributed way whilst protecting privacy. So we want to do that. How often are the data updated? We have hundreds of different data flows. And it depends on the data flow. General practice data during COVID, we were getting monthly, but we mainly do quarterly because it's just more efficient for us to do it. The hospital data we get monthly, but it's always a couple of months behind when they get their coding. During COVID, we had the COVID test results on a daily basis. So we can manage to automate and set up things depending on which suits people, but a lot of it is very up to date.
Thank you, Mick, Jardine. Fantastic talk. Thank you so much. My interest is a little bit more of the end user researcher, perhaps, and the interest in uptake and adherence to guideline indicated medications. I think we're all a little bit jealous of the setup you've got there, but we do have sort of, ad hoc linked data sets here. And I know I've worked with Jana on one of them. In that setting, it is quite useful to do parallel analyses because those implementation questions can be quite specific to the setting. We've had ad hoc relationships with the Swedish group about doing parallel analyses there. So the question with that long preamble is, how do we actually enable collaborations with this broader group, or is it a bit of an ad hoc relationship between individual researchers?
Yeah, so probably the easiest answer is send me an email and I will put you in contact with people who do similar things. And then hopefully it'll progress from there. I mean, we do a lot of analysis on different things. We don't do performance management for named by general practice or hospitals like that. So when we were setting this up, actually, that was one of the things they didn't want to see. They said they get endless amounts of performance management, and the last thing they wanted was more and more of it in the back of that. So we do de-identify the practices and the hospitals and all that, but you can still do the pattern work.
Yes, and that's not the interest so much. It's more about the underlying drivers, not individual practices, which is temporary phenomenon. It's more what are the predictors, what are the overall system and individual drivers. So
and a lot of it's really interesting. I mean, there were slides I cut out because there were too many about what the impact of COVID has had on our treatments for cancers and lots of other things. And we're involved in a European project looking at that. And you can see different effects in different countries. Some countries barely affected at all. You think, wow, fantastic. Particularly people in Finland didn't seem to make any great difference. Whereas I think for us, like we had major differences with early cancer treatments. Now they've come back to normal, but our joint replacement stuff is still way down from where it is and where it is in many other countries. So lots of these, I think, become very interesting in cross-jurisdictional analysis.
Yes. And just the sort of analyses where we're comparing effects in different jurisdictions and tell you the impact of health system structures.
Yeah.
And it's a good example, isn't it, that not just unmet need, but unprovided need.
Yes.
Yeah. So I was wondering about the trauma link in the first COVID bit and the first COVID lockdown. And we obviously come into work with policies in place for work health safety, but we don't do work health safety very well in our own homes.
No.
So was it just a quantum effect of people at their homes where there is an increased frequency of accidents, but also that horrible spectre of domestic violence that also happened?
There will be some of that, because that was something that was clearly obvious in the data and in the police data. So there has been an increase in that. There's not so many fatalities involved in that in our system. So I think a lot of this will be older people falling. And it may be they've just become deconditioned, they've got less support. There will have been fewer people coming into houses to help with them because everybody was a bit on their own. The UNSW people were wondering about whether there was a lot more home injuries and DIY stuff as well. So people got bored, everybody is locked away. They started doing things like painting roofs and going up trees and all sorts of things which they normally wouldn't do. And there may be some of that.
I think I would also suggest, though, it's the frequency of people taking soup upstairs to a COVID positive person, and they were doing that 4 or 5 times more than they would otherwise do. And it's just little things like that that happened in the home that just increased trauma.
I mean, it's something we just need to find a little bit of time to have a look at, so to try to understand what went on.
And then, Ronan, maybe the last question then, if everyone's finished. Oh, one more over there.
Nick Buckley here. We have something here a bit similar, the Shore. And one of the things that people complain about most is, you know, you decide to change an axis by 10% and it takes 2 weeks to get the new graph out of the system. What's your turnaround, and do you have any ways of kind of actually making it more iterative, getting things out?
I think our turnaround is like 48 hours. So what we tried, I mean, this is one of the difficulties, is that we try to teach people not to look for too much out, right? So you basically want to take out what do you need for the paper or the presentation, rather than actually I've created 500 pages of results and I want to take it all out because I don't want to do it. The problem is you have human eyes that need to look at all of those things. So the more you ask for them, the more of a queue that it builds up. So persuading people and training them in the first place about what is and isn't allowed out. And all of our people who are on this have to have verified training courses. I had to do mine the other night for the Office of National Statistics on statistical disclosure to be allowed to continue to do it. But persuading people about actually this is what you can and can't get out. Some types of things are much easier for the reviewers to see. Regression coefficients and all that tend to have a problem. You know, frequency tables tend to often have problems for suppression of that. So it's a bit about training people, but we do turn things around in 48 hours.
Okay, so I think this is the last question now before we have something to eat. And that is, how many of those studies that you've presented today has actually gone into changing policy and into implementing new clinical pathways?
Oh, a lot. There's a lot of this. I mean, I could have gone through lots of different ones. I mean, we had a natural experiment around air pollution alerting, which was interesting. So, you know, like, okay, pollution is bad for people. So the government says, stop producing pollution. Then they don't really do anything to do that. So the next thing is to tell people who are vulnerable to actually avoid pollution incidents. All right, so they've got forecasting systems in London that. Well, we've got a steelworks down the road, and they got funded at one stage for an almost instantaneous pollution alerting system around that system. And that came along, and we said, oh, it'd be interesting if this is possible to evaluate. So the local GPs said, yeah, we'd be interested in that. So they recruited a whole pile of people who would sign up to this sort of alerting system, and there was a whole pile of controls, match controls who didn't. Now the concept, everybody said, well, okay, people will stay away from the pollution, they'll get less sick, and they'll be less busy in the hospital. Now, what did happen was during the 2 years, some of those people had 200 pollution alerts, who produced lots of pollution. Part of those alerts also said if they were feeling unwell to consult their practitioner. In the intervention group, we had twice the number of emergency department attendances and 4 times the number of admissions. So it basically worked exactly the opposite way it was intended to do. And fantastically, we decommissioned the service. So there are various examples like that, both of positive and negative policy impacts I could go through.
So further questions? I think we'll now go and have a few drinks and a few canapé. And I just want to thank Ronan for a wonderful presentation and its wonderful, impactful sale database. So Thank you for Ronan.
You're very welcome. And I gotta say, the fact that we've done it means anybody can do it. And I think it's a good example with your own policy leads and saying, we're not putting anybody's data or health at risk by doing this. So if it's possible to do in Wales, it should be possible to do in New South Wales and elsewhere.